Good day everyone. So today we're here to talk about the ester band and urea cycle. To start off, let us take a look at the definitions. So, what is ester band? An ester band is a covalent bond formed when the hydroxyl is a group of the carboxylic acid and hydrogen atom from the alcohol condensed with the removal of water molecule. The general formula of the functional group of ester is negative CORR. So when we say ester bond, an ester bond is a chemical linkage formed between an alcohol group and carboxylic acid group through a process called a condensation reaction. This reaction results in the elimination of a water molecule, creating a new bond that connects a carbon from atom and the carboxylic acid to an oxygen atom, which is then connected to another carbon group from the alcohol. The bond that forms between the oxygen and carbon atoms is then known as an ester bond or ester linkage. Many everyday products such as cosmetics, flavoring agents for beverages, and industrial cleaning agents contain ester. While these are usually formed in a lab, esters also occur naturally. How ester bonds form? An ester bond is created when the hydrogen atom from the carboxylic group, which is the negative COOH of our carboxylic acid, is replaced by an alkyl group, which is the R, from an alcohol in a process called acidification. This results in the general formula RCORR for esters, example of ester bond formation. So, one common example is the reaction between ethanol, which is an alcohol, and acetic acid, which is a carboxylic acid, that form ethyl acetate. A type of ester the reaction is, and there are the formula. So, a type of ester. There are different types of esters, including carbonic, phosphoric, and sulfuric esters. Carbonic esters are the most common, with ethyl acetate being the first identified. And lastly, ester bonds are defined as chemical linkages, which is the negative COO, that connect extended carbon chains in organic compounds, particularly in esters, which are known for their stability and high latent heat storage capacities. So now, let's proceed to chemistry of urea cycle. So, what is urea cycle? Urea cycle, also known as the ornithine cycle, is a cycle of biochemical reactions that produces urea NH2 from ammonia NH3. Animals that use this cycle, mainly amphibians and mammals, are called ureotelic. The urea cycle converts highly toxic ammonia to urea for exertion. So, the urea cycle is a filtering process to remove toxic substances from your body and keep other substances that are good for your moving throughout your body. Urea, which is the carbamide, is a substance made by your liver. The urea cycle begins when you eat. Urea begins when you eat, so the urea cycle is like a cleaning system in your body. Its main job to get rid of toxic stuff. Think of it as a filter that catches all the bad stuff and lets the good stuff keep flowing. Why is the urea cycle also called the ornithine cycle? Understanding the ornithine cycle is also known as the urea cycle. It is a series of biochemical reactions that convert ammonia, which is toxic, into urea, which is less toxic and can be excreted from the body. So, What is another name for the urea cycle? The urea cycle or ornithine cycle converts excess ammonia into urea in the mitochondria of liver cells. Another name for the urea cycle is the ornithine cycle. And lastly, urea cycle is a series of 6 reactions necessary to rid the body of the nitrogen generated by the metabolism, primarily of amino acids from the diet or released as the result of endogenous protein catabolism. Arginase is the sixth and final enzyme of this cycle. And I think that's all. Thank you.